Well, thanks everyone anyway. So today we have special guest, Marija Markicevic. Is that correct, Marija?
Almost. Markicevic.
Okay, not almost. You were being very...
It's okay.
Markicevic. Oh, I've typed it many times, but maybe never said it aloud. Okay. Well, special guest and friend of the lab and group and co-collaborator and all-round good scientist and nice person, Marija. And she's going to talk about some of her work that she did in collaboration with Valerio Zerbi, Nikki Wenderoth, and others at ETH Zurich, and also during her visit here to Sydney with some of us. Thanks, Marija, for agreeing to talk to us. Marija currently has COVID.
That was a main secret.
Oh, is that a secret? Okay, Marija is currently completely fine. We'll cut that out of the recording. Well, it's good to have a little. You were a big trooper but she says she's fine and wants to give the talk anyway. Well, thanks, Marija, for taking the time and preparing, and looking forward to the talk.
Well, thank you, Ben, for the invitation, and thanks everyone for joining in. So as Ben has said, this is work that has been done during the end of my PhD and part of my postdoc at ETH Zurich, together with Nikki and Valeria, that were my PIs, not collaborators. So this is where we try to actually use Ben's HCTSA toolbox to understand how ball dynamics is shaped after cell-specific D1 neuromodulation. And this is going to be very interdisciplinary talk, so please stop me at any point to clarify anything or that is unclear. It's going to be easier that way. So now the motivation, let's see. Yes, where does this all come from? So we know that brain is a multi-scale complex system that consists of millions of perpetually interacting neurons that they all together form microcircuits and ultimately brain networks. This is the structural foundation that underlines any healthy brain function. And in order to fully understand this brain function, it is necessary to define these relationships across all these different levels. And in today's modern science, there are a number of ways to do so. But what is one modality that is particularly important for this talk and this work today is functional magnetic resonance imaging, or more specifically, resting state fMRI. We know that resting state fMRI from the last 30 years of research is actually based on slow oscillations in the blood oxygen level dependent signal, which is known to display coherent activity in the absence of any tasks. We also know that it is non-neuronal. It does not come directly from neuronal activity. However, there is tons of evidence that BOLD has also it has origins in neuronal activity as well. And one of the recent works that comes from my current host lab at Yale shows that calcium imaging, the networks that come out of calcium imaging data, which is direct neuronal activity, and BOLD signal from fMRI do overlap to quite some extent. So we know now that there is more recent evidence that there is neuronal origin in the BOLD signal. And I will move on to say that the motivation for this work that I will present today is as this figure comes from a recent opinion paper from Emily Finn and Mac Shine, which I am pretty sure you guys are familiar with, where they argue how fMRI can be really used as a catalyst to integrate different subfields of neuroscience. And what this work does is tries to, by neuromodulation at the macroscale level, bridge across scales to try and understand how this cellular level neuromodulation is displayed at the network level. And we do that by using resting state fMRI in a mouse, together with chemogenetics, and we apply a highly comparative time series analysis to analyze this data later on. Now, before I dive into the methodology of the work, a brief intro into chemogenetics. So in order to manipulate a mouse brain, we need to inject a viral vector of interest into the mouse brain, into the region of interest in the mouse brain. That viral vector holds a fluorescent tag, which is mCherry, which allows us to track it later on when we want to see where it actually transfected, and a dread, which is, in this case, a type of neuromodulator that we used. It's actually a modified G protein coupled receptor, which once it's activated with a small dose of clozapine, it can cause a chain of events that can either cause excitation or inhibition, depending on the type of the dread that we decided to use, excitatory or inhibitory. So this is the neuromodulator that we used in this study. And now what did we do with it? So we perturbed the activity of D1 medium spiny neurons within the dorsomedial striatum. So for many of you, I'm sure, are familiar with the direct pathway, which works as follows. Once the striatum receives excitatory inputs from the cerebral cortex, the striatum reacts to it by activating a direct pathway, which means D1 medium spiny neurons are activated and send inhibitory projections to globus pallidus internal and substantia nigra pars reticulata, which further on disinhibit the thalamus and allow some action to happen. So within this very well-researched and very well-defined network, we neuromodulated only those medium spiny neurons that are located within the dorsomedial striatum. And we wanted to see, by using fMRI, what happens to the dynamics within this well-defined system. Now, what is the exact methodology that we followed? So after the viral expression of the dread, which allows us to excite these neurons, we've placed the mice in the box with a light and a camera and recorded it for 25 minutes, 10 minutes after activating the clozapine, after activating the dread with the injection of clozapine. And a week later, so that they can recover from this experiment, we performed a resting fMRI experiment where they were scanned for 38 minutes. The first 15 minutes were a baseline. At the 15 minutes, we activated the dread by injecting the clozapine and then continued with the recording. And now, before I go into the resting state results, which are the main interest, I guess, of this audience, I will go into I will explain a bit, how did we actually know that we targeted D1 medium spine neurons and that we actually excited them as we said we are doing. So there are two ways that we double checked for that. First one is by tracking the location of the viral transfection, with this mCherry tag that I've mentioned in a few previous slides. So this is an overlap of all the animals that underwent that were injected with the excitatory dread. And to be a bit more specific, for each animal, we've checked three things. First, we stained for DAPI is a chemical that stains for DNA nuclei. Then mCherry is a fluorescent tag that allows to target where the dread is. And prodynorphin is a D1 marker, which means it's only found on the D1 medium spiny neurons. And this final product here means that, yes, we've actually targeted D1 medium spiny neurons with our DREAD. And here is another one that shows that there are a few here, and they show that we actually were successful in activating it with the DREAD with the clozapine using CFOS targeting. So now that we know that we are successful with the location and the targeting of D1 medium spiny neurons, here are the results of behavior. So as you know, we unilaterally excited the D1 medium spiny neurons. And we know from literature that that should cause the mice to increase in their controversive rotations. And that's exactly what all our mice that had an excitatory DREAD expressed. This is a video that is 4 times its original speed. So it's been speeded up. The mice don't really rotate that fast, but I think it's very clear from it that there is an increase in the controversive rotations. And we have quantified this for every animal in our experiment, and there is indeed an increase in controversive rotations and also an increase in total distance traveled. So now we know for sure that we are doing what we say we're doing, and that is neuromodulating D1 medium spiny neurons within the dorsomedial striatum. And so from now on, I'll move on to fMRI results. First, starting with a very traditional functional connectivity results, where before I tell you what they are, I have to highlight that in order to, so we only assessed regions that form corticostriato-thalamic loop. And in order to obtain these regions, we used the Allen Atlas and the significant connections defined by the Allen Atlas. And these are the regions here, green are the cortical regions and blue are the thalamic regions that we that came out of this Allen Brain Atlas system. And what you see here in this plot is a basic connectivity before any neuromodulation. And as you can see, there are some pretty strong, basic connectivity between dorsomedial striatum and the rest of the regions. And as you can see, there are some pretty strong and significant connections as anatomically expected. Now, what happened when we activated the DREAD and performed the same analysis is we saw that in anterior cingulate, retrosplenial region, and motor cortex, there is a decrease in functional connectivity in the D1 animals that received the D1 excitation compared to controls. And that is the only significant result and significant change in the long-range functional connectivity that we could see. Now, after this, we've moved on to what is, for me, the most exciting part of this paper is the feature-based time series analysis. That is a part that was done together in collaboration with Ben, where we've used his highly comparative time series analysis toolbox, which combines and leverages thousands of time series features across different disciplines, such as ones that deal with correlational structures, static distribution, stationarity, like sliding window measures, basic function, and many, many, many more things. There are over 7,000 features. These are just a few to just place the flavor on the table. Now, what I did with this toolbox is I extracted the time series from each region of interest in that striato-thalamo-cortical loop, and converted it to a features to appropriate features using the toolbox, and then performed a classification analysis using tenfold cross-validation with the SVM and 50 repeats. For each, so within each region, within the striato-thalamo-cortical loop, balanced classification accuracy was calculated, and then, of course, corrected. So now I will, this was, of course, done for the entire 30 regions that formed this loop. How I will proceed to present the results by splitting them into striatal, cortical, and thalamic regions, because it's easier for interpretation. But what is important to note here is that all the statistics, the FDR corrections, and the multiple comparisons, all these things were performed on the entire 30 regions together. So not just within the striatum or within the thalamus. It's just, so that is clear. Now, what did we see in the targeted striatal regions? So here we have, in the literature, there exists a mouse corticostriatal projectum, which is a very good tracer study that splits the striatum, which is a very complex region, into 29 different subregions. We've used this knowledge in these subregions and combined it together with our viral expression maps in order to, for each animal to choose where did the virus transfect, or did the virus transfect a certain region of this projectum or not. And based on that, we put together two regions, one called CP virus, that's where the viral transfection was, and another called CP other, that's where there was no viral transfection detected. And we asked, then we performed the balanced classification accuracy analysis and saw that there is a significantly altered dynamics within the region where we injected the dread with the viral expression, and not in the other, region as expected. Now, I will move on to the thalamus. In the thalamus, when we assessed the ball dynamics within a large number of different regions, what came out immediately is that there is a set of regions with very high classification accuracy versus others that don't show any changes after D1 excitation. And at first, we didn't know why these regions were special, but after a very thorough research, literature research, specifically in anatomical and tracer studies, what we found out is that all these regions, thalamic nuclei with statistically significant changes in ball dynamics after D1 excitation, are actually anatomically connected to our neuromodulated region, not just by receiving inputs from it, but also by projecting directly back to it. And that's not true for any of these other thalamic subregions. So, and now for the cortical, for the case of cortical regions, here after D1 excitation, we saw only four regions that show very highly, significantly altered ball dynamics. And those are the primary and somatosensory and motor cortex, visual cortex, and the association area. What was clear from it is that only those regions that are unimodal, that means they receive projections, lower number of projections and integrate less functions compared to transmodal, showed a significantly altered ball dynamics. Now, this unimodal to transmodal hierarchy gradient was obtained from work from Ben, from what he did in this work is he ordered cortical areas along a putative functional hierarchy from unimodal to transmodal using these diverse sets of measurements such as cell density, cytoarchitecture, gene expression, and so on. And so we obtained the rankings from this work and ranked our balanced accuracy results. And what we saw is a very, very strong correlation between the two, which indicates that D1 medium spiny neuron excitation alters the cortical area's ball dynamics with a strength that follows a certain hierarchical position, with the strongest change in unimodal and the weakest change in transmodal regions. Now, the last set and the next intuitive thing that we ask, and the last set of analysis that we performed was, so now we know these 10 regions that displayed significantly altered dynamics across cortex and thalamus and striatum, but what exact features and what exact dynamics change after this D1 excitation? And for this is a dendrogram of only one significant region, and that is one thalamic significant region. So how we assess this is we determined which individual time series feature differs significantly between the two groups, between the controls and the excitation group, by calculating a Mann-Whitney rank sum statistic. And of course, that was calculated for each feature within one region. And then it was FDR corrected for all features. And in this FDR correction, we had a number of them that survived this FDR correction. And here we have top 100 of them that are ranked by how similar their behavior is, which was calculated using absolute Spearman correlation. And here is displayed as a Spearman correlation distance. So we see different types or families of features, which are denoted by different colors, and how similar they are, specifically here to the autocorrelation feature, because it's a very intuitive feature. We know what increase or decrease in autocorrelation means. And here we show that exactly this feature in ventromedial thalamus and laterodorsal thalamus after D1 excitation shows increases in autocorrelation, which means that broadly in these two regions, there is a D1 excitation leads to slower and more autocorrelated fluctuations. Now, you may ask, but what about all the other cortical and thalamic regions that displayed significantly altered ball dynamics? Well, for them, when we calculated Mann-Whitney rank sum statistics, for each feature, and FDR corrected that, we could not find any significant differences. But what we did, regardless, is we were broadly interested whether these non-FDR corrected Mann-Whitney statistics are in any way similar to how correlated they are to these two regions that display very significantly Mann-Whitney feature scores that are very significant. And what we found out is that when we correlated the feature scores between the ventromedial thalamic nuclei region and all these different regions, we found a correlation that's relatively strong, which means in broad terms that after D1 excitation, all these other regions also lead to slower and more autocorrelated fluctuations. And with that, I'd like to conclude and say that in this study where we excited D1 medium spiny neurons of dorsomedial striatum and performed a classification-based analysis using features, we show that ball dynamics are shaped by anatomical connectivity of thalamic subregions within the neuromodulated dorsomedial striatum. They're also shaped by the spatial location of the cortical regions along a certain putative hierarchical dimension. And broadly, all these significantly altered regions show that their dynamics becomes more predictable by increasing their autocorrelation structure over time. And with that, I hope I showed you how fMRI can be used to bridge scales and different subdisciplines of neuroscience. And I'd like to acknowledge everyone who has been involved in this study and also my current hosts at Yale University. And thank you for your attention. If you would like, the access to the paper is on this QR code. And yes, I'm welcome for any questions. I haven't heard any. So, oops, that was not planned.
Thanks, Marija.